Hi, welcome to the Bioinformatics Chat. As usual, I'd like to start this episode by telling you about the war in Ukraine. However, I'm acutely aware that there is now another major war that's going on, and it pains me to see so many people suffering in Israel and Gaza at this very moment. My story today is about Mariana Mamonova, a combat medic of a Ukrainian Marine Infantry Battalion. Mariana survived 6 months of Russian captivity as a prisoner of war while being pregnant, until she was exchanged and freed in September 2022. I hope that hearing Mariana's cheerful voice might give a bit of hope to those of you who need it right now. These are just a few excerpts from a 2-hour-long interview that I will link in the show notes. I was at our medical post on the seashore. Our commander contacted us via the field telephone and said, "Expect landing of Russian Marines from the sea. Defend the shore." What? What landing? Are you serious? There's 5 of us, 5 assault rifles. Actually, it was very scary. Never mind my laugh, I was really scared. At that time, I was already pregnant, but I didn't know about it yet. I just didn't have any time for myself. Everyone was taking care of someone else. I had to think about what to do with the wounded, how to retrieve them, how to get them to the hospital, since the road was being shelled. I found out in the middle of March, when I was at the steel plant in Mariupol. I had a single pregnancy test, which I found by accident while going through my stuff. It was like being in a reality TV show when they suddenly appear and tell you, "Congratulations, you're going to be a mom." And you're like, "Oh my God, no, this is not happening to me." They're talking to someone else. And they're like, "No, it's you. Congratulations." Aerial bombs are falling, everything rumbles, and you get the news that you're pregnant. People ask me why I didn't tell anyone. What would it change? I'm the only medic of my unit, of my battalion. What should I have done? Just leave? Leave everyone? I'm an officer and I'm a medic. For me, it was unacceptable. I'm a Marine. For me, it was unacceptable. So I made a decision. I would stay here and I wouldn't tell anyone. In the interview, Mariana describes how she ended up as a POW and what happened to her in captivity. Fortunately, they didn't beat me. Yes, they humiliated me, but you swallow it. You don't show it. It is unpleasant. For a woman, there are many things that men say that are unpleasant. You digest it and you say to your child, it's not directed at you, it's towards your mother. Because not everyone in this world is good. You have to understand that. That's how it is for now, but over time things will improve for us. Until the 6th month of pregnancy, I had hoped that they would release me. It got really scary in the 9th month when I was taken to the hospital in the occupied city of Donetsk. They told me that there were no exchanges because the 2 sides could not come to an agreement. I was very round and due soon. I was very afraid to give birth there. Still, I kept telling my child, it's all temporary. We will come back home. You're strong. Finally, Mariana talks about her exchange and release. They take away your blindfold and you see Ukraine, Chernihiv region, big yellow letters. People tell you, "Congratulations, you're home now." Seriously? Home? Congratulations, you're in Ukraine now. Do you understand what kind of word it is? Ukraine?
В Україні, розумієш, яке це слово? Україна, дома.
And the most important thing they tell you, you're a free person now. Just a free person that can do anything. All this Chernihiv border, it's all yours. And no matter in which corner of Ukraine you live, it's all yours. I walked and I cried. After 6 months of being captive, I think a lot of people who come back, they fall on their knees. I was falling, but they caught me. I thought I would fall down and roll like a little bear. Today, Mariana is well, and shortly after her release, she gave birth to a healthy girl named Anna. Unfortunately, there are still many more POWs in Russian captivity, whom Russia would not exchange or provide any information about. And now, on to bioinformatics. My guest today is Marie Sadler. Marie is a PhD student at the University of Lausanne, and she actually is going to defend her PhD thesis just a few days after this episode is being recorded, which will happen by the time you're listening to this episode. So feel free to go and congratulate her. Hi, Marie. Welcome to the podcast.
Hello, Roman. Thanks for having me today.
Your work that we will be talking about today is, there's a paper in Cell Genomics called Multilayered Genetic Approaches to Identify Approved Drug Targets. And can you talk about your background and how it led you to this work?
Sure. So originally, I did not start in the field of bioinformatics or genetics or biology. I actually studied chemical engineering, but I was always more attracted to the computational aspects of any class we had regarding computations or mathematics. So during my master's, I then reoriented myself towards data analysis and bioinformatics, and that's how I then started a PhD in statistical genetics with Zoltan Kutelik at the University of Lausanne. And so the focus of my whole PhD thesis is how can we integrate genetics and medication to better understand disease mechanisms that we could target with medication, but also treatment mechanisms, like how does the drug interact in the body and how could we identify deviations from expected pharmacological response, which then also goes into the field of pharmacogenomics.
And so regarding this particular paper, right, what was the main goal of the study?
So previously, studies have found that the drugs who target genes that have a genetic evidence in the population that gene is linked to the disease through natural variation, that these drugs are more likely to be approved. And now over the years, there's many different methods due to the emergence of different data sets that allow us to establish this genetic evidence. And there wasn't really any systematic study in the field yet that compared them side by side to see, is it more useful to only look at GWAS data? Can we gain any further insight when we integrate QTL data, like omics data, to have this intermediate information of the transcript or protein that's mediating the genetic effect? Or now with the advent of sequencing data, do we gain even further information by having rare variant information and their genetic effects on diseases? So the goal was to compare all of them in the most fair manner, to see objectively which method performs best.
And so when you say genetic support, what exactly do you mean by that?
So basically, if we have a gene and individuals may carry genetic variants within that gene region that make them more susceptible to that disease or even protect them against that disease. So this means that this gene is having a causal role in conferring disease risk. So from genetics data, we have a very strong evidence about the causal gene, which we may not get from other data sources. So it's basically a huge, large clinical trial everybody's participating in by having information about their genetics and the disease to find these causal genes.
So do you consider drugs and diseases that have only the genetic cause, or is it also like, do you consider drugs that may like target some protein and you link that drug to the gene that encodes the protein without necessarily there being like a variant in that gene that causes the disease, but just in order to alleviate the disease or cure the disease, you have to target this protein? Is this also like a link you consider?
We have to distinguish between two things, that what we try to find with the genetics are causal genes or causal pathways, but the people who develop the disease and whom we treat with the drugs that we found by looking at individuals carrying the genetic variant, they may not have the disease because of the genetics, but the drug still works for them. So for instance, for LDL cholesterol, PCSK9 inhibitor is a very famous example where rare variants in that gene were found to protect against hyperlipidemia and heart diseases. But it's only a very small number of individuals who actually carry these variants in this PCSK9 gene. But now the recent studies that have been done on antibodies, but also RNA silencing molecules, they found that these drugs are very effective in the global population, where the majority of the individuals, they do not actually carry a genetic variant in that gene or protein.
Right. So the genetic variants, they just help you to establish that link.
Exactly.
And so if you imagine like a drug development pipeline, what stage of this pipeline are you targeting? On which stage are your findings most applicable, I guess?
So they're most relevant at the very early stages. So when it comes to find the target. So finding the target, and then later we can have these big screens or specific protein design that can inhibit the target that we identify. So it would be at a very early stage. Now, is genetics also relevant for later stages, clinical trials, whom to recruit, at least at the beginning, to already have earlier signs of maybe if we enrich the clinical trial with a population for whom we know it works better based on their genetics, can it be helpful? But this research does not cannot be applied to that. So we're not there yet, I think, to really define ideal clinical trial populations based on the genetics, at least not to my knowledge.
And so if I understand correctly, the rough process that you suggest or you have in mind is that, so we have a disease, and regardless of whether that disease has some genetic causes or not, but maybe we could try to find like a GWAS that links that disease with some genes, right? And then we look at the genes, we look at the gene products, and we consider those gene products as potential targets for our drugs. Is that roughly correct?
Yes, exactly. Yes.
And so walk me through the different prioritization methods, right? So they differ by what data they get as inputs. So some are based on GWAS, some are based on GWAS plus some additional studies, right? Like pQTL and eQTL, and some are based on exome sequencing. Walk me through these different methods that you consider and you compare to each other.
Right. So the first method we used was based on GWAS data. So we used a statistical method called Pascal, which is able to attribute gene scores to a GWAS summary statistics.
Just in case not everyone is familiar with what a GWAS is, maybe let's start with that.
Yes. So in a GWAS, we compare very large populations of individuals who are either healthy or have a disease. In that case, we are in a binary setting. And in a genome-wide scan, we test each genetic variant, whether the allele dosage, so whether a person is homozygous on the reference allele, heterozygous, or homozygous on the alternative allele, and we correlate that with the disease status. So this gives us a genetic effect size and also genetic strength or p-value to see how strong that effect is or how statistically significant. We can repeat the same thing in a quantitative setting where our disease is a continuous variable. For instance, LDL cholesterol, where we don't classify people into high or low LDL cholesterol level, but we have a quantitative value. Again, we get a genetic effect size and a p-value. So this GWAS analysis, they can be done on genotyped data. So on a microarray, you typically genotype about 1 million variants, and you can impute up to 80 million variants. Not sure we have to go into the details of that, but basically we have genetic effect size for roughly 10 million variants throughout the genome. And these are rather common variants with allele frequency above 1%. So this data input is used for the GWAS method, or we call it GWAS method in the paper, where we use GWAS summary statistics and also an LD reference panel to get the local correlation structure on the SNP level to calculate gene scores. Because in a GWAS, we have a SNP genetic effect size, so effect size on the variant level, but we want to have an effect size on the gene level, which aggregates multiple SNPs around the region of the gene.
At this point, this is purely correlational, right? And so when we talk about effect sizes, it's not that we're claiming that there is a direct effect of the SNP on the disease, but this is just some somewhat arbitrary number, right? That established like the correlation degree.
So given that we're working with genetic data, we can almost certainly claim that's causal, so that we do have the direction that the gene is causing the disease. There can be some confounding if that's what you're referring to.
Well, I mean, first of all, as you say, there's this linkage disequilibrium. That means that a gene can show this effect even though it's a completely different gene causing the disease, for instance. Or one SNP versus a different SNP, like this particular SNP could not be the causal one.
Exactly. So we're not yet sure what is the causal SNP. We do have a likely causal signal, but finding the causal SNP or the causal gene, that's where we use this method, like Pascal. But the causal SNP may not actually be measured in our GWAS data. And as you say, the local LD structure, from there it may be hard to find which one is the causal SNP and which SNPs just correlate with the causal SNP, and we still find an association. So that's the main issue, actually. If we have multiple genes in a region that are potentially overlapping and we have a signal there, like to which gene do we attribute that signal? So what's the causal gene? And we use gene scores, but again, we may still be wrong about which gene is actually causal, given that so many genes are overlapping and all will get a very high genetic score. So GWAS has been, like in other studies that looked at the genetic evidence of drug target, have been, let's say, the most common or popular method to use to establish genetic evidence for potential drug targets, because GWAS, they have been conducted now over 15 or 20 years almost, I think. And the summary statistics that you get, you can easily share them. So we no longer have individual level data. So in that sense, this is a really great method, because now in the GWAS catalog, you have so many thousands of summary statistics that you can use to identify causal genes. But then again, as you mentioned, we may not know what is the actual causal gene, and we also may not know about the mechanism. So even if we find a signal in the GWAS, we may not necessarily know by what it is driven. Is it because of a protein coding variant? Is it regulatory? And also, if it's regulatory, in what direction? Is it like increased gene product causing the disease, or decreased gene product?
Okay, so I think it's pretty clear with the GWAS, with the genome-wide association study, but then you also propose or consider integrating that with something called eQTL and pQTL. Can you explain what those are and how do they help?
Yes. So as I mentioned, with GWAS, we may still be unsure about the causal gene. Therefore, over the years, many large-scale omics QTL data has been generated, where large transcriptomic studies have been conducted to find the genetic effect on the transcript level. So whether genetic variant is increasing or decreasing expression levels. And the same has been done on the protein level. So now if we overlap our signals that we have from the GWAS with the QTL data, we may be more confident in finding what is the actual causal gene by looking at the transcript or the protein product. So for this, we were using then the largest eQTL study that has been conducted so far from eQTL Gen, where transcripts have been measured in whole blood and meta-analyzed across different studies. GTAX is another very great consortia that we use because there we actually have transcriptomic information on, where we have eQTL information available across different tissues, around 50 tissues, which is of great use because the genetic effect size on the transcript level may vary whether we look at the brain or in the blood or in the liver. So in this data set, the sample size is, however, a bit smaller, given that it's harder to get all of these tissues. And for protein data, the largest studies so far have been done in plasma. So there we were using data from the DECODE study where protein levels were measured in up to 35,000 individuals.
So if I understand correctly, the difference between the non-integrated GWAS and the GWAS integrated with, let's say, eQTL, and that will probably relate to what we will be discussing later regarding the whole exome sequencing. There it's sort of different, but in the GWAS, most of those variants are regulatory, so their impact is not through the affecting the protein structure, but through regulating some other genes, right? And so the difference between the pure GWAS and GWAS integrated with eQTL is just the way you attribute, to which gene do you attribute a particular SNP. So in GWAS, you more or less simply find the closest gene to this SNP or closest genes. And in eQTL, because you have this information of which SNP affects expression of which gene, instead of simply looking up the closest gene, you go to that other gene that, so this SNP affects, let's say SNP A affects the expression of gene B, and then you attribute, you can more accurately attribute the effect of SNP A not to some closest gene, but to actual gene B, for which there is evidence that SNP A regulates gene B.
Yes.
Is that roughly correct?
That is a very good summary. Yes, thanks.
Good. And I guess it's the same for pQTL. Is there what is the conceptual difference here? Obviously, apart from being based on different data between eQTL and pQTL, and maybe I don't remember if you explained the difference. So eQTL means it's based on measuring the transcripts, right? And pQTL means it's based on measuring the protein levels. But how would you describe, on the conceptual level, maybe the implications of using different types of data?
So conceptually, they are the same, the method. It's literally just what is measured. So for eQTL, we measure the transcriptome, and there we have a pretty good coverage of almost all protein coding genes. And in the pQTL GWAS method, we measure the proteins. And their current high throughput methods do not yet capture the whole protein coding space. So we are more limited in the number of proteins that we can assess. So we were using data that were using the SomaLogic platform, and there roughly 5,000 proteins were measured. And we do have significant protein QTL, so significant effects or genetic effects on only about 1,800 proteins. So the space at which we can look at is much smaller than when we did the eQTL GWAS analysis. And also regarding the tissues for the eQTL GWAS, we looked at about 50 tissues by having the GTAX consortia data, whereas for protein QTL GWAS, we only had information from plasma proteins. But the way the data is integrated is exactly the same.
So then, a priori, what would there be any advantages to looking at proteins instead of the transcripts, given this lower throughput?
So in theory, looking at the protein level, given that drug targets up to now are mostly proteins, it would give us the advantage that what we observe on the transcript level is actually translated onto the protein level. So in theory, we would be more confident if we see a protein trait link that this is actually a good drug target candidate than if we only see a transcript trait link.
Right. Okay, and then there is the third or the fourth method, depending how you count, which is based on the exome sequencing. And can you explain what that is and how it works?
Yes, so for the exome method, we used actual sequencing data. In this case, we used the whole exome sequencing data that was generated in the UK Biobank. And as already said by the name, we're looking at protein coding variants, and more specifically at very rare variants that you can only capture through sequencing method, as opposed to genotyping method that up to now mostly used for GWAS analysis. And the way this method works is that we concentrate on these very rare variants. In our case, we use a threshold of variants with a minor allele frequency below 1%. And we also concentrate on only the variants for which we expect that there is an impact on the protein level. So those that are predicted to be either missense or putative loss of function. So this missense or putative loss of function variants that are very rare are collapsed into a burden. So each person gets a burden assigned, and that's correlated to the trait. So either the quantitative trait or the disease healthy status, or disease status, sorry.
Shall we explain in more detail what those things mean? So what is missense and what is putative loss of function?
Yeah, so missense is that when we do have a different allele, this may have a protein coding effect or not, given that amino acids, you can have the same one with different sequences. So not all of them are actually protein altering. And putative loss of function, those were really predicted by algorithms to actually severely or alter the function of the protein. So there we're actually more confident that this variant results in a protein that's no longer fully functional. So this was a choice. You could also only look at putative loss of function variants or only look at missense variants. A previous paper, Bachmann et al. 2021, where they look at the whole UK Biobank, they found that the statistical power was increased when you combine both types of variants.
And what is the rationale for only considering the rare variants? Like what would you lose if you also included the not so rare variants in this study?
So if you then also include the not so rare variants, you're more likely to recover the GWAS signals, where you look at common variants. But the GWAS, like what's the advantage here is that you may pick up genes that are more constrained in the population. So for whom you don't have common variants with the variant disease link. Well, I mean, it's still ongoing research for many people, I believe, but there's a bit of consensus. I feel that if you have very high sample sizes, both methods may converge. Like you find the same disease genes if you look at GWAS or at exome, like the whole exome sequencing data in like doing this burden tests. But if you're having lower sample sizes, you may have different genes, which is also what we find in our study, that the genes prioritized by the GWAS, they're not necessarily the same as those prioritized by the exome method, because you may find these more constrained genes that you only find by setting this minor allele frequency to very low thresholds, and that GWAS may miss.
And so what did you find regarding these four methods? Which one was the best?
So yeah, as I already mentioned before a bit through the data that goes in, they are not necessarily comparable the way you would ordinarily use them. For GWAS, you would go and use the largest GWAS you find, which is usually a meta-analysis between different cohorts. Whereas for the exome method, we only have access to the UK Biobank at the moment from where we have summary statistics of this gene burden tests. So we did all sorts of adaptations also with respect to the number of testable genes. As I mentioned before, for pQTL GWAS, we could only test 1,800 genes because we did not have pQTLs for the others, and the number of measured proteins is currently only at about 5,000. So we then conducted our GWAS and QTL GWAS methods on GWASs only from the UK Biobank to have a fair comparison with the exome method. And we also downsampled the number of background genes that we tested to make it comparable with the pQTL GWAS method. And when we then made all of these adjustments, we found that there was no difference between the GWAS and the exome method, and both were the highest ranking methods. But GWAS did perform significantly better than both QTL GWAS methods.
Oh no.
Yeah, I know.
we're integrating, right? We're adding more data, and it actually becomes worse. Is that what you're saying?
Yes. So at first it doesn't sound very satisfying. Like, why would you spend all of this money on generating this large QTL data sets? I mean, at the same time, it's not necessarily surprising, because with the current data that we use, other people have shown that this QTL data only mediates a fraction of the disease heritability. So as this one big study, Yao et al., I think 2020, where they showed that only about 11% of the disease heritability goes through or altered gene expression levels based on GTEx data. So 11% is really low. So what happens is that many GWAS signals we cannot explain by current data sets because we may miss the right context. We have the largest data for whole blood, but not all of these disease-causing genes, their causal tissue may not be blood, it may be somewhere else. And in GTEx, we are currently rather underpowered to get this genetic effect sizes on transcript levels in all these other tissues. So that's a big point. And also, well, this may not explain it entirely, but we only use Mendelian randomization to do this integration of QTL and GWAS data. There's other methods, although I don't think if you would use another method, you may drastically improve the yield.
And can you explain briefly what Mendelian randomization is?
Right. So that's Mendelian randomization, as we were discussing before, allows you to get the causal effect or putative causal effect of an exposure, which in our case is the transcript or the protein level on the trait on outcome, which here is disease or disease-relevant phenotype. And you use instrumental variables to get this causal effect, which for us is QTL data, which is the genetic effect on the exposure, and then the GWAS data, which is the genetic effect on the outcome.
And so what you're saying is the eQTL or pQTL methods did not perform so well, but you think it's not because of the conceptual issues with the methods, but just because of the limitations of the data we currently have?
Yes, that's correct. But what is also important to say is that even though they did not necessarily perform so well, they do provide us more insights than the other method. Like, we are really confident that if we find an effect, it's really because increased gene product or decreased gene product is causing the disease. So when we think about drug targets, we want to know, do we have to develop a drug that's inhibiting the function or rather an agonistic drug? So in that sense, they are still very valuable, although they were not as performant in identifying this known drug targets. And for the exome method, while we are almost sure that it's because of the loss of function of the protein that we observed the disease, we cannot be entirely sure. It could be that the variant is actually trait increasing or trait decreasing. Like, it's not entirely obvious from the burden statistics in what direction it goes.
And by the way, we didn't also explain like what this burden is or how it's calculated. Is it just a sum of the scores of like individual variants, or what is it?
So there's different ways to calculate the burden. The way we used it, we used actually the summary statistics from Bachmann et al. So we calculate the burden ourselves. But it was like people who would not have any rare missense or putative loss of function variant, they would be encoded as 0. So their burden would be 0. And if individuals had at least one of these variants, it would be 1. So even if they had 10, I don't think it's realistic, but if they had, let's say they had 10 rare variants in that gene, they would still be 1 unless one of these variants, they were homozygous on those. So the individuals who had both copies of their gene or the variants predicted to be rare missense or putative loss of function, they would be 2. And then this burden is then correlated with the disease status. But there's different ways. You could also use the sum, so really counting how many of these rare variants an individual carries and then correlate that with the disease status.
And maybe now is a good time to explain how you actually evaluate the methods. So the methods, first of all, what do they output? Does each have to prioritize a fixed number of genes? Do they each have like 10 attempts to guess the gene, or do they produce different numbers of genes? And how do you then judge whether that prioritized set of genes is a good one?
Right. So for each method, we get a gene p-value. So that's our starting point on how we get this gene prioritization scores that we overlap with the drug targets. And what we do then is, to be as fair as possible, we do not set a threshold on the p-value that we get, but we take the top 1% genes as the significant disease genes. Except for the pQTL GWAS method, we used 5%, given that our gene universe was only about 1,800. Using 5% would result in a more comparable number of diseased genes with this 1% that we used for the other methods. So we used 2 different approaches. One was using a Fisher's exact test to really get an enrichment value between this prioritized disease genes and our drug target genes. And the other approach was using the area under the receiver operating curve, where we don't need to set a threshold. So there we don't say it's 1% genes, but we look at the whole distribution to get an AUC value.
But in both cases, you rely on the sort of ground truth set of genes. And where does that come from?
So there we use different databases to derive what is actually a drug target gene and for which disease is this gene a drug target. So we use Campbell, where we get this drug indication and drug protein link. Then we also use from a rather recent publication, Ruiz et al., the drug indication links. We use the Stitch database that provides us drug to protein interactions, and the GGIDB, the drug gene interaction database, that also gives us this drug gene interaction link. And we combine these databases to then go from the indication to the drug, and then from the drug to the gene to get this target indication links, which is what we are interested in. I mean, in our benchmarking analysis, we are not looking on the drug level. There can be many drugs targeting the same gene, and we would only be interested in the gene-disease link. And we decided to look at different databases because not all of them define drug targets in the same way.
Combined together, how many drugs would you say does that data cover?
So what we find is that on average, the number of drugs that we have per disease is around 40. But then the number of targets really differs a lot, or the number of, gene-disease links. If you look at the Campbell database, we had a total of about 700 gene-disease links, whereas in the other databases, it was more about 7,000, because these databases, they would report many more drug-gene interactions than what you have in Campbell.
So generally, do they define these links as the drug, the molecule interacting with the gene product, or what are the definitions that they use?
Yeah, generally, I think it's more the gene product. And what's the main difference is that they scrape the information from different original sources, and then they combine it all together. So there can be some contamination when the gene is rather a pharmacogenomic gene, so maybe it influences drug concentration in the body, but it does not have an effect on the drug efficacy.
Okay. And for scoring, do you consider each of these sources independently, or do you just combine all those genes in a one big set?
So we look at these sources or combination of sources independently. And then we also give a combined benchmarking score where we combine them all. And we do take into account how correlated they are when establishing the confidence measure that we have on the score.
And so one aspect of your study that we haven't mentioned yet is the use of networks and network propagation. Can you talk about that?
Right. So if we have a drug that's targeting a gene, the effect of the drug on that gene may be propagated to nearby gene that then results in the expected pharmacological response. So what this also means is that the disease genes that we find, they may not be direct drug targets, but through these gene interactions, they are related to a drug target. And we try to test this hypothesis by doing network propagation. So here we used 3 different networks. The first one being the string protein-protein interaction networks, and the other 2 being co-expression networks. So starting from our initial gene prioritization scores that we got from these 4 different methods, we used a Markov random walk algorithm to propagate that signal on the network. So that interacting genes may suddenly also become diseased genes. And even more if these are neighboring genes of highly prioritized diseased genes by any of the 4 methods.
And so since you have like a particular, you said like top 1%, right? That means that after network propagation, some genes that were prioritized will be no longer prioritized, but instead they would sort of yield their place in the list, in this short list, to other genes.
Yes, absolutely. So the 1% top genes, they will be a bit shuffled around. Yeah.
And then presumably you compared whether that gives any benefits, right? If it's more sort of accurate, and was it?
So yes, it was. So network diffusion helped in identifying known drug targets. The analysis became a bit more problematic when we looked at the string protein-protein interaction network, because there it helped so much that just looking at the network and not at all at the initial disease genes was the best predictor of drug targets. So what I mean by this is what we found is that the node degree, so to how many genes a given gene is connected, that was the best predictor whether that gene was a drug target. So these hub genes, which is likely because drug target genes are generally more studied. So in the string PPI network, where literature is also included, these are hub genes, and therefore we have this kind of circular argument that what we put into our algorithm is already defined by what is a drug target, and then we benchmark on drug targets. But for the unbiased co-expression network, it also improved a bit, but by far not as much.
Yeah. And so this brings me to this thought that what you're trying really to predict, or what you are actually predicting, is the genes that would be classified as drug targets of approved drugs versus the probability that a given drug will be approved given that it targets or doesn't target a particular gene, right? So it's somewhat different sort of metric, and one does not necessarily lead to the other.
Right. Well, I guess this can be seen more from a machine learning perspective if we do an out-of-sample testing of whether this gene is predicted then in the future to be a good drug target, or whether we just try to predict what has been known in the field, if I got it correctly.
Yeah, and I'm thinking more like from a Bayesian perspective, right? So we have like 2 different conditional probabilities. There is the probability of gene being a target given that the drug was approved, and the probability of drug being approved given that it targets a particular gene. And we don't necessarily have the data to transition from one probability to the other using the Bayes rule. So it's like a prediction in a different direction, not predicting whether the drug will be approved given what it targets, but predicting, given that it's approved, what will be found as a target of the drug.
Yes, it's more the second thing, because historically it was not necessarily known what would be the drug targets. Like very often these have been found later on, and now retrospectively we do find that they were enriched for this genetic evidence. But this has changed now. Like if you look at the more recent drugs, there was a good prior knowledge that this gene is causal, often, or I think now pretty obviously through genetic evidence and large-scale genetic studies. So this type of analysis, doing them on recent drugs, would give a very different result.
So do you have any thoughts on the future of this? Are there any like new approaches that you can think about?
Yes. So what we found in the study is that the drug targets prioritized by the GWAS and the exome method, they were often very different, suggesting that we get complementary information from GWAS or exome. Although, as I alluded to before, is that as sample size increases, both methods, they may actually converge to the same gene. But given that as of now, the information may be complementary, it may make sense to combine the scores from both to get, let's say, a more perfect combined score. And also for the GWAS method, so far we used a very, well, let's say, simple algorithm of just using GWAS summary statistics and an LD reference panel to calculate a gene score. But there's much more information on the SNP level of whether this SNP is regulatory, what's its function. So all this functional annotation data could be integrated into getting a more accurate genetic score and have a better mapping of what is the causal gene if we do have a very gene-rich region. So those are different avenues that could be looked at in the future to get a better combined gene prioritization score. And then, of course, when it comes to networks, as I was showing in the study, we were showing that the string network is very biased, and the co-expression networks that we used, they did improve the score, but not as much. But again, here context may be important as well. And when we do have a disease where, let's say, the liver plays the major role, maybe it makes more sense to have a co-expression network based on liver data and not whole blood or any other tissue. So again, having a more precise context and based on that, deriving the network structure maybe help to get good gene prioritization scores.
Well, Marie, thank you very much for coming to the podcast. It was very interesting to talk to you, and good luck with your defense.
Thank you very much.